Identification and manipulation of bacteria through biochemical and growth testing requires an array of techniques and specific tests. Two of these will be demonstrated here. The spread plate technique is used for enumeration of bacterial cells in solution. Using aseptic technique, a tube containing a diluted culture of cells is mixed thoroughly, and a small volume is added to the surface of a sterile plate. Quickly, before the liquid can be absorbed into the plate, a spreader, which can be made of glass, plastic, or metal, is sterilized by dipping it in alcohol and setting it on fire. After the alcohol burns off, the spreader is allowed to cool. Check that the spreader is not too hot by placing it on the plate surface away from the culture. This will also help to cool the spreader as the heat is transferred into the plate. Now, spread the culture to all edges of the plate while rotating it to get an even distribution of the cell suspension. After allowing the culture to absorb, the plate is inverted and incubated to allow colonies to form. After incubation, this plate can be counted to get a viable cell count. SimDeep Agar is a multi-part testing system. The media takes the form of a semisolid deep, a tube of low-concentration agar that allows cells to migrate through it. To inoculate this deep, a wire needle is straightened out and then sterilized. The wire is placed into the inoculating culture and pulled out. It is now coated with cells. The wire is aseptically stabbed straight down into the agar and then pulled straight out to try and avoid slicing into the agar. If the stab is straight, it will be easier to observe motility the next day. After incubation, a SimDeep will be scored for sulfur reduction. A positive result is observed in the form of a black precipitate. You may be able to determine the oxygen requirements for the culture by observing the areas of growth and whether or not the culture is motile by looking for a cloud of growth away from the stab. A few drops of Kovacs reagent is added to the cultured tubes to check for indole production. Within seconds, you should observe the solution turning magenta for a positive result, or remaining colorless for a negative result.